So the focus of my research here at WPI is in understanding the causes and consequences of aneuploidy in human cancer. So many of you in the audience are probably familiar with the idea that each of the cells within your body contains 46 chromosomes. So this is 23 chromosomes from your mom and 23 chromosomes from your dad. And that would look something like this. So in order to ensure that each of your cells have the same number of chromosomes, there's a series of checkpoints that the cell goes through to make sure that each time a cell prepares for division, they're going to make identical copies of each of these chromosomes and then sort them and ensure that they're segregated equally into the two resulting daughter cells. And again, this is essential to make sure that all of the cells in your body have the correct number of chromosomes. However, on occasion, defects in this process do occur, and this results in daughter cells that have an abnormal number of chromosomes, known as an aneuploidy. So when these changes in chromosome content happen early in division, they can impact human health. One of those examples is represented here on the bottom, where these are the chromosomes from a person with Down syndrome. And what you'll notice circled in red is that this person has 3 copies of chromosome 21 instead of the expected 2 copies. When these changes in chromosome copy number occur early in development, if they don't cause lifelong impacts to human health, they can result in viability issues. And we now know that changes in chromosome copy number are the primary cause of miscarriage during pregnancy. We see even more extreme cases of aneuploidy in cancer, and that's what my research is focused on. So we know that over 80% of solid tumors display some degree of aneuploidy. That's represented in these 2 images here, where on the top we can see a single cancer cell that has 3, 4, or even 5 copies of some chromosomes, while the representation on the bottom is a cancer cell that has a couple of chromosomes that contain only a single copy. So my research is focused on understanding how cancer cells are able to generate these complex aneuploidies and what their impact is on tumor cell survival and the care that the patient should receive. And so the reason why we're interested in aneuploidy is we know now that the presence of these complex aneuploidies can impact the way a tumor cell responds to therapy. So if you'll imagine that each of these colored circles represents a single cancer cell, and each change in color represents a division where a segregation error in chromosomes has occurred and generated a new aneuploidy. What you'll see is in just a few cell cycles, we very quickly not only see the growth of the tumor, but see an increase in this heterogeneity, where the different colored cells have different genetic content. And this means that they have the potential to respond differently to therapeutics. So when this patient receives chemotherapeutic treatment, although it's efficient at killing many of those cancer cells, this aneuploidy enables some of these cells to resist therapy, to remain proliferative, and now these cancer cells can regrow and repopulate the tumor environment. And in doing so, they are now repopulating a tumor that maintains that drug resistance. So this is a clinical challenge. So what can I, as a cancer cell biologist, do to address this? Well, research in my lab is using genetic engineering and imaging-based approaches together with molecular and cellular biology to try to understand how chromosome segregation is corrupted in cancer cells. We can make movies shown here of cancer cells going through cell division, both in the presence and absence of chemotherapeutic agent, and ask questions like, how are these cancer cells different than their normal counterparts? We can look carefully at the chromatin structure itself, at the molecules that are involved in associating these chromosomes with the spindle machinery that's responsible for segregating those chromosomes. And in doing this, we're learning about those molecules and pathways that are essential for the generation of aneuploidy in cancer. And using this knowledge, we can attack cancer in 2 distinct ways. So we can use this knowledge to prevent these segregation errors from happening, and in doing so, potentially limit the ability of cancer cells to evolve and adapt to therapeutic treatments. And we can also use our understanding of how these cancer cells are different from their non-aneuploid counterparts to specifically target those cancer cells for cell death without disturbing the normal non-aneuploid cells around them.